GCT members and future scientists, my name is Annie Etuk and I am very fortunate to work as a postdoctoral research fellow in Professor Kasim's group here at the UCL Great Ormond Street Institute of Child Health. Our work focuses primarily on developing cell-based therapies for the treatment of leukemia. Now, I've been asked to give a brief introduction to CRISPR and gene editing, so hopefully by the end of this little talk, you will be able to discuss the basics of DNA, genes, and mutation. There'll be an overview of what CRISPR is and how we have adapted this bacterial system to enable gene editing in the lab. This will be followed with a talk by my esteemed colleague, Giorgio Ottaviano, and he will be discussing a specific example of our research and how gene editing tools like CRISPR can be used to create universal donors. He will also follow up with some information on ethical and safeguarding considerations of our research. Now, so that you can see all of the content of my slides, I am going to turn off my camera. The cells in your body contain a complete set of genetic material that you have inherited from your parents, which we refer to as your genome. Now, the genome is squashed into little structures called chromosomes that you may often see mapped and arranged like in this picture. Chromosomes consist of DNA, which is made up of building blocks that we call the DNA bases. You can think of the DNA in your genome as an instruction manual that makes you who you are. Your DNA is split into sections that we call genes, and these genes code for characteristics such as your height and the texture of your hair. Genes are also translated into proteins that can influence how certain cells develop and how they function. Now, sometimes there are mistakes in this instruction manual, mutations in gene DNA that can potentially lead to disease. Now, these mutations could be inherited, spontaneous, or caused by a mutating agent such as UV. Now, the mutation may translate to an altered form of a protein that no longer works properly. And if this protein is essential for cell development, then the cell may not survive, or alternatively may proliferate out of control like we see in cancer. Now, scientists can determine what mutations cause disease and what changes to make to ameliorate these conditions, and that's where CRISPR comes in. Now, the CRISPR-Cas system is a tool that scientists can use to edit or change DNA within a genome. CRISPR-Cas was discovered in bacteria and is part of an inbuilt immune system that protects bacteria from invading viral DNA. Now, this bacterial immune system was adapted for use in the lab by a number of scientists, including recent Nobel Prize winners and all-round science queens, Jennifer Doudna and Emmanuelle Charpentier. Now, the system we use in the lab comprises two components: a small molecule of RNA that acts as a guidance system and is therefore referred to as CRISPR guide RNA. We like to think of guide RNA as a sat-nav homing in on a specific DNA postcode. The second component is an enzyme called Cas9 that can cut strands of DNA like a pair of molecular scissors. Now, together, these two components form the CRISPR-Cas complex, and when the CRISPR guide RNA finds its target, i.e. a section of DNA that matches the guide RNA, it locks onto that specific section and slices both strands of DNA. This cut can then be exploited in order to edit what was originally there and therefore correct a mutation. Now, there are gene editing platforms that predate CRISPR-Cas, such as TALENs and zinc finger nucleases. They are also two-component systems. However, in these earlier editing platforms, the guiding and cutting components are fused together, making them complex and expensive to synthesize and requiring a new fusion protein for each mutation that you want to target. So dare I say it, the sexiest thing about CRISPR-Cas is that the guiding and cutting elements are separate, meaning you only have to customize simple pieces of RNA to guide you to the mutation and then provide this common Cas enzyme to cut at that targeted location. So CRISPR-Cas is faster, easier, and cheaper than what came before. So, targeting and cutting with CRISPR-Cas means that you can introduce a break in DNA at a specific location and exploit this break to insert, delete, or modify genetic sequences. And that is the fundamental principle of gene editing, a DNA break followed by some form of correction. But when you introduce a break in DNA, the cell will try to repair the damage, and there are two mechanisms or pathways that can be triggered when DNA is broken. In the first pathway, the DNA is cut, the gene is broken, and does not stick back together properly, essentially disabling the gene. Now, a broken gene means a broken protein, so we can use this pathway to prevent protein production or, for example, to turn off or knock out a receptor. Conversely, in the second pathway, Cas makes the cut, and at the same time, we provide a new piece of DNA to patch and repair the gene sequence. This way, we can introduce a precise gene modification. Moreover, the CRISPR-Cas system has now evolved to allow what we call base editing. So with slight modification to the Cas enzyme, we can now precisely substitute a specific DNA base. Now in this example, we have swapped the DNA base from a C to a T, much like the find and replace function you can use in a Word document. So there you have it. In summary, we have covered the basics of DNA, genes and mutation. We now know a little bit more about CRISPR and how this system has been adapted to enable gene editing in the lab. And in a short while, you will learn about the clinical applications of this technology with an example of how gene editing platforms like CRISPR can be used in our research. Before I hand you over to Giorgio, I'd first like to acknowledge Professor Kasim and all of my wonderful colleagues in Team Wasim, and of course the variety of funders who make our research possible.